Good morning, students. Chapter number 5, Principle of Inheritance and Variation. Let us continue with the next topic, linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid cross carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied, white-eyed female to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that two genes didn't segregate independently of each other, and the F2 ratio deviated very significantly from the ratio obtained in the dihybrid cross by Mendel over peas, which was based on that the two genes segregate independently. Morgan and his group knew that the genes were located on the X chromosome, that is one of the sex chromosome, and saw quickly that when the two genes in the dihybrid cross were situated on the same chromosome, the proportion of the parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage. मॉर्गन की स्टडीज़ रिवील करती हैं कि बहुत सारे जीन्स जो कि पास पास एक ही क्रोमोसोम पर प्रेजेंट होते हैं, उन केस में उनका सेग्रीगेशन डिफ़िकल्ट होता है. फिर उन्होंने लिंकेज टर्म कॉइन की डिस्क्राइब करने के लिए कि किस तरह जीन्स जो हैं वो फिज़िकली एसोसिएटेड हैं किसी भी क्रोमोसोम पर. एंड ऑल्सो दे कॉइन द टर्म रिकॉम्बिनेशन टू डिस्क्राइब द जनरेशन ऑफ non-parental gene combinations. Morgan and his group also found that even when genes are grouped on the same chromosome, some are very tightly linked and showed very low recombination. In figure, it is shown as cross A, while others were loosely linked that showed higher recombination. In figure, it is described as cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombination between gene pairs and on the same chromosome as a measure of the distance between the gene and mapped their position on the chromosomes. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing project described later. Let us try to understand the cross done by Morgan in Drosophila melanogaster. As stated earlier, he used two crosses. In one, he took yellow and white genes and other wild type. He obtained wild type as well as parental type in F1 generation, whereas in cross B, he took white miniature against wild type and obtained wild type and white miniatures. But the percentage differed in both the crosses. In first cross, parental type was 98.7%, while in second cross, it was 62.8%. In the first cross, the recombination types were 1.3%, while in second cross, it was 37.2% in the gametes. Then he selfed the F1 generation and seen the F2 generation and obtained all the four genotypes. And the ratio deviated from the Mendel's ratio 9:3:3:1. This cross, we can locate the genes very easily. In the cross A, they are closely linked, while in cross B, they are loosely linked on the same chromosome, that is X chromosome. Polygenic inheritance. Mendel's studies mainly describe those traits that have distinct alternate forms, such as flower color, which are either purple, or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in human, we don't just have a tall or short people as two distinct traits, but have all the possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume that 3 genes A, B, C control skin color in human, with the dominant forms capital and recessive forms small. Capital responsible for dark skin color and recessive responsible for light skin color. The genotype with all the dominant alleles will have the darkest skin color, and that with all the recessive alleles will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color, that is capital A, capital B, small b, capital C, small c. In this manner, the number of each type of allele in the genotype will determine the darkness or lightness of the skin in an individual. मतलब जितने ज़्यादा डोमिनेंट एलेल उतना ज़्यादा डार्क कलर, जितने रिसेसिव एलेल उतना लाइट कलर. Similarly, pleiotropy. We have so far seen that the effect of the gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene. The underlying mechanisms of pleiotropy in most cases is the effect of gene on metabolic pathways which contribute towards different phenotypes. One of the example is the cause of disease phenylketonuria, which occurs in human. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. Single gene mutation occurs here. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. That is, the same gene is responsible for both the functions. Next is sex determination. The mechanism of sex determination has always been a puzzle for the geneticist. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of bases for sex determination. Henking could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperms received this structure after spermatogenesis, whereas the other 50% sperm didn't receive it. Henking gave a name to this structure as X-body, but he couldn't explain its significance. Further investigations by other scientists led to the conclusion that the X-body of Henking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of XII type, that is, all eggs bear an additional chromosome, X chromosome, besides the other chromosomes. Other chromosomes are known as autosomes. On the other hand, some of the sperms bear X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome becomes female, and those who didn't fertilize became males. Do you think the number of chromosomes in male and female are equal? Due to the involvement of X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of XII type of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes beside the autosomes. These observations led to the investigation of number of species to understand the mechanism of sex determination. In a number of other insects and mammals including men, XY type of sex determination is seen, where both male and female have the same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is not X but the Y chromosome, that is smaller than the X chromosome. Females, however, have a pair of X chromosomes and they both bear the same number of autosomes. Hence, the males have autosomes plus XY, while females have autosomes plus XX. In human beings and in Drosophila, the male have one X and one Y chromosome, while females have a pair of X chromosomes besides the autosomes. In the above description, you have studied about two types of sex determining mechanisms, XII and XY. But in both cases, male produce two different types of gametes, either with or without chromosome, or some with X and some with Y chromosome. Such types of sex determination mechanism is designated to be example of male heterogamety. In some other organism, example birds, a different mechanism of sex determination is observed, as seen in the figure, the third one, C. In this case, the total number of chromosome is same in both male and female, but two different types of gametes in terms of the sex chromosome are produced by the females. That is female heterogamety. The chromosomes are named Z and W. In order to have distinction in the mechanism of sex determination described earlier, the two different sex chromosomes of female bird has been designated to Z and W chromosomes. In these organisms, the female have one Z and one W, whereas male have a pair of Z chromosomes besides the autosomes, as seen in the earlier two methods of the sex determination in the organisms. Sex determination in human. It has already been mentioned that sex determining mechanism in case of human is XY type. Out of 23 pairs of chromosome present, 22 pairs are exactly same in male and female. These are autosomes, while the last pair of chromosome is the pair of sex chromosome, in which a pair of X is present in female, while presence of X and Y are determinant of the male characteristic. During spermatogenesis, among males, two types of gametes are produced. 50% of the total sperm produced are with X chromosome and the rest are with Y chromosomes besides the autosomes, that is 22 plus X and 22 plus Y. Females, however, produce only one type of ovum with X chromosome, that is all the female gametes are 22 plus X. There is an equal probability of fertilization of ovum with the sperm carrying either X or Y. In case of the ovum fertilizes with the sperm carrying X chromosome, the zygote develops into female, and the fertilization of the ovum with Y chromosome carrying sperm results into the male offspring. X sperm fusing with the ovum gives female, while Y sperm fusing with the ovum gives male. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is fortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. जबकि ये बिल्कुल क्लियर है कि स्पर्म डिसाइड करता है कि द चाइल्ड विल बी मेल और फीमेल. A unique sex determination is there in honeybee. The sex determination in honeybee is based on the number of sets of chromosome an individual receive. An organism formed from the union of sperm and egg develops a female which may be a queen bee or a worker bee, but an unfertilized egg develops as a male that is drawn by means of parthenogenesis. This means that the female consists two sets of chromosome while the males consist one set of chromosome, that is, females are diploid while males are haploid, that is, they have 16 chromosomes. This is called haplodiploid sex determination system. It also consists a special characteristic feature, that is, the male produces sperm by mitosis. However, we have studied that gametes are always produced by meiosis. They do not have father and thus cannot have son, but have a grandfather and can have grandsons. Here is the representation. Parents female have 32 chromosomes. They undergo meiosis and produce gamete with 16 chromosomes, while male have 16 chromosomes and they produce gametes with mitosis containing 16 chromosomes. The male gamete fertilizes with the female and form females, while males are produced by the unfertilized eggs of female. इसीलिए कहा कि मेल के पापा नहीं होते और वो कभी दादा भी नहीं बन सकते. So this was all about sex determination in different organisms, the three types of determining methods XII, XY and ZW. Remaining topics we will cover in the next lecture. Till then, keep studying.